Okay, so for chapter 4, enzyme. Okay, so these are the subtopics of chapter 4. We are going to look at catalysis and concept of activation energy. So you already know basically what is activation energy. Okay, symbolized by EA. And then mechanism of enzyme action. For this one, we are going to look at lock and key hypothesis and induced fit hypothesis. And then next is we are going to look at factors affecting the rate of enzyme reactions. So basically there are 4 or 5 factors. Okay, so you have the temperature, you have the pH, you have the enzyme concentration, and you have the substrate concentration, and also the presence of inhibitors. Okay, so those are the factors that will influence the rate of enzyme reactions. Okay, so next we are going to look at Michaelis-Menten kinetics and also Lineweaver-Burke plot. So these for these two, basically in this subtopic we are going to look at a Michaelis-Menten constant Km. Okay, what is the significance of you finding the value of Km and also Vmax. Okay, so in order for you to understand this subtopic 4.4 Michaelis-Menten, you have to understand the concept that you learn for the factors affecting enzyme reaction, especially on substrate and enzyme concentration. Okay, so when you understand how enzyme rate of reaction is influenced by enzyme concentration and substrate concentration, then you will understand this subtopic of 4.4 Michaelis-Menten kinetics and Lineweaver-Burke plot. And then next we are going to look at cofactors. So basically cofactors are a non-protein component of enzyme that the enzyme requires to help the enzyme to catalyze reaction. Okay, so some enzyme requires cofactors, some don't, but most of it requires cofactors to assist, okay, in the enzyme reaction. Okay, so these cofactors, there are several different cofactors based on how the cofactor helps the enzyme reaction. Okay, and then next we are going to look at enzyme inhibitions. So for this one, we are going to look at competitive inhibition, non-competitive inhibition, a fuse inhibition that will affect enzyme activity. And then 4.7, regulation of enzyme activity. This one, in this subtopic, we are going to look at some mechanism also relating to, for example, cooperativity, allosteric regulations, okay, feedback inhibitions. So those we are going to look at in this subtopic. Okay, and then 4.8, classification of enzyme. So enzymes are classified into 6 classes, if I'm not mistaken, 6 classes based on how the enzyme catalyzes the reaction. Okay, so there are enzymes that involve in hydrolysis, okay, such as maltose, okay, like lactose, maltase, lactase, macam itulah kan. So enzyme that involve in breaking of bond using water. Okay, so those are group of class of enzyme. We group them under hydrolase, for example kan. So hydrolase, enzyme that involve in breaking of bonds using water. So itu salah satu dia lah, hydrolase. Okay, so kita ada oxidoreductase. So ada 6 lah, lyse, okay, lysis. So so we are going to look at the 6 classes of enzyme based on how the enzyme catalyzes the reaction. And then last 4.9 is enzyme technology. So we are going to look at how enzyme are used in the industry in large scale. Okay, macam tu. Okay, using method we call it as immobilized enzyme. Okay, what is immobilized enzyme and the benefit of using immobilized enzyme. Okay, So these are the learning objectives. So explain how an enzyme lowers the acquired activation of energy for a reaction. So a reaction, when it is catalyzed by enzyme, it will lower the activation energy and speed up the rate of reaction. Okay, so without enzyme, the reaction can still occur, but it will take a longer time for the reaction to occur because the substrate or the reactant needs to absorb as much as heat energy, for example, heat energy that is from the surrounding to catalyze the reaction. Okay, so without enzyme, the reaction can still occur, but at a slower rate. Okay, so that is the significance of having enzyme in the reaction, to lower the activation energy and to speed up the rate of reaction. Okay, and then second, explain the mechanism of action and describe the hypothesis relating to its mechanism of action. So for this one, we are going to look at like I mentioned, the lock and key hypothesis and also induced fit hypothesis. Ada dua lah kat situ kan, lock and key and induced fit. Okay, induced fit mechanism, ada dua. Okay, so explain the formation of enzyme substrate complex. Okay, so for this one, as you know, every enzyme has its own specific substrate to catalyze a reaction. So once the substrate enters into the active site of the enzyme, it forms the enzyme substrate complex before it turns or it reach the transition state. Okay, transition state to produce the product. Okay, so explain factors that affects enzymatic reaction. So saya dah mention tadi, you have the pH, temperature, substrate concentration, enzyme concentration, and also the presence of inhibitors can also affects rate of enzymatic reaction. Tapi yang kita akan concentrate for the first four adalah yang tu tadi lah, enzyme, concentration, substrate concentration, pH, and also temperature. Inhibitor kita akan tengok lagi spesifik later in this chapter. Okay. And then explain the double reciprocal plot of enzyme kinetics. So this one is related to the Lineweaver-Burke plot tu kan. So it is related to Michaelis-Menten kinetics and also Lineweaver-Burke plot. Okay, the significance of you knowing what is km, what is the value of km, and also Vmax. Okay, so from this reciprocal plot, you can determine these two values. Kan? Kenapa nak tentu sangat these two values? Kita akan tengok nanti. Okay. So list types of cofactors. Okay. So cofactors, there are mainly three types. Okay. So you have the coenzyme. Okay. You have the metal ions or activator. Okay, and then you have the prosthetic group. Okay, prosthetic group. Okay, so those are the three different types of cofactors that assist. Okay, assist ataupun help lah, can help or assist in the enzyme reaction. Okay, discuss the mechanism of enzyme inhibition. So the presence of inhibitor in an enzymatic reaction can affect the enzyme reaction. So the inhibitors, can either bind to the active site, okay, or another site of the enzyme. We call it as the allosteric enzyme. Okay, so awak dah familiar dengan active site, okay, but for additional detail of this chapter that is different from what you have learned in the school, you have another site in the enzyme. We call it as the allosteric site. So for example, here you have an enzyme. So usually, okay, awak tahu active site, or sorry, all enzyme have active site. So there's actually enzyme with another site we call it as allosteric site. Okay, so substrate will bind to the active site. Okay, so you have another molecule that binds to this allosteric site that affects the enzyme reaction. So the presence of this allosteric site will allow either inhibitor to bind to the allosteric site or activator to bind to the allosteric site. Okay, so obviously if the inhibitor bind to the allosteric site, it inhibit the enzyme reaction. So if activator binds to the active site, it activates the enzyme. Okay, so kita akan tengok apa jadi nanti kan, what will happen if the inhibitor binds to the allosteric site to the of the enzyme. Activator apa jadi? Okay, what will happen if the activator binds to the allosteric site? Okay, so which is next? The next LO, learning objectives, is for you to discuss how inhibitors or activators regulate enzyme activity in cell. Okay, so inhibitors can bind to either active site or allosteric site. Activators bind to the allosteric site, okay, to activate the enzyme. So list classes of enzyme and explain its role. Tadi lah macam saya cakap tadi kan, if I'm not mistaken, there are 6 classes of enzyme depending on how the enzyme catalyze the reaction. Okay, so discuss the importance and the main techniques of immobilized enzyme. Okay, so in the industry, you will use this immobilized enzyme to maximize the production of the products, okay, without affecting the enzyme concentration, okay, without compromising with the enzyme structure or concentration in the reaction. Okay, macam tu. Okay, so we are going to look at for the first subtopic, which is catalysis and concept of activation energy. So as I go along the notes, okay, my note will be a little bit different from yours, as I will add as I've added some extra information. Okay, so if you think that what I've added is important, so you may jot down later, okay, as we go along the lecture. Okay, so catalysis and concept of activation energy. So characteristics of enzyme. So most enzyme are globular proteins. So this one we have looked at in chapter 1 for proteins. Okay, so you can classify protein based on its structure. Okay, so proteins can be globular proteins or structural protein, kan? Okay, fibrous protein, globular or fibrous. For enzyme, we group them as globular proteins. Okay, so usually for enzyme, they will attain the tertiary and the quaternary level of protein though. Okay, tertiary and quaternary of protein to gain this globular shape of the protein. Okay, so enzyme has to fall into specific shape so that this specific shape will form a specific shape of the active site, so that specific substrate can enter into the active site. So there has to be globular, and furthermore, globular protein they are soluble in water. Okay, soluble in water. So why does the enzyme needs to be soluble? Because enzyme are found in your body fluids, are found in your cells. Okay, so those environment are in aqueous environment. Okay, so enzyme will catalyze reaction in this aqueous environment. They need to travel in your blood, for example, from the organ that secrete and produce the enzyme. Okay, so they needs to be soluble. Okay, so that's why most enzyme are globular proteins. Okay, so they act as catalysts. Okay, they act as catalysts, which is a chemical agent that speed up a reaction without being affected by the reaction. Okay, so or if the enzyme are found in living cell, we call it as biological catalyst lah, and biological catalyst. So characteristics of enzyme that you all know, that it is not consumed or changed in the reaction. Okay, so berapa banyak awak letak enzyme dalam reaction tu, macam tu lah dia ada before and after the reaction. Okay, so the enzyme will not be consumed or the structure of the enzyme will not be changed. Okay, as long as you make sure that the pH or the temperature of the enzyme is in its optimum condition. Okay, so if you change the pH, if you change the temperature, you can change the structure of the enzyme. Okay, so it can lead the enzyme to be denatured, for example. Okay, so substrate binds to the active site of the enzyme. So the enzyme, every enzyme is highly specific. Okay, highly specific. Okay, specific. So it means that every enzyme will have their own substrate to catalyze certain reaction. Okay, certain reaction. So this is due to the presence of the active site. Okay. Okay, so the shape of the active site will only allow certain substrate to enter into the enzyme. So that's why enzyme is highly specific. Okay. So the enzyme, because it is not consumed, it can be used over and over again. Okay, it can be reused. Okay, it can be reused over and over again. So those are the characteristics or the property of enzyme that you have to know. Okay. So so for the explanation, so enzyme, okay, enzyme when it catalyzes the reaction, it will lowers the activation energy barrier, ataupun we write the symbol as PA. Okay, lowering the activation energy barrier. So and reaction that is catalyzed by enzyme, okay, will lower the activation energy, but without the presence of the enzyme, the reaction can still occur, but at a very slow rate. Okay, at a very slow rate, and the reaction will require a higher activation energy. So as the substrate needs to absorb as much as thermal energy as possible from the surrounding. Okay, thermal energy from the surrounding. Kalau tak ada enzyme, aktiviti reaction tu akan still berlaku, tapi pada kadar yang sangat rendah disebabkan oleh activation energy yang diperlukan adalah lebih tinggi. So energy ni datang daripada the surrounding. Okay, thermal heat, thermal energy ataupun heat energy. Macam tu. Okay, so by having enzyme in the reaction, so what are the benefit is that it brings the substrate together in the correct orientation. So as you know, the enzyme will have active site. So when you have two or more substrate to bind to the active site, so by having the enzyme, it makes sure that the substrate are oriented in the correct orientation for the reaction to occur. Kalau tak ada enzyme ni, substrate tu boleh berdebaran, bersepah, macam mana nak reaction tu berlaku? Faham tak? So by having the enzyme, so it allows the substrate to come together in a correct orientation by binding at the active site. Macam tu. Okay. So number 2, provide a favorable cellular microenvironment. So what does it mean? It means that by having the enzyme, it allows a certain microenvironment to be achieved. Okay, local environment surrounding a cell, which is by having the active site, it provides a more conducive environment for the reaction to occur. Macam kita tengok tadi kan, the presence of cofactor. So enzyme that has cofactor in that enzyme structure, it helps the enzyme to assist the reaction. So this cofactor can make the environment more suitable for the reaction to occur. Okay. So number 3, stretch the substrate bonds towards their transition state. Okay. So transition state is achieved when the substrate binds to the active site, and at this state, okay, the bond within the substrate will starts to break. Okay, we start to break and start to form new bonds. Okay, that is transition state. Okay, so the transition state is achieved and allows the bonds to be broken. Okay, it causes the bond of the substrate to be in a stress condition. It bends the bond of the substrate so that new bonds can be formed. All bonds are broken, new bonds are formed. Okay, to produce the product. Number 4, form temporary covalent bond with the substrate, which is directly involved in the chemical reaction. So enzyme, okay, by having the cofactor, for example, this cofactor can within the enzyme can directly involve in the reaction. So contohnya kalau substrate tu dia nak break bond, for example, it involve for example the transfer of electron, okay, from one substrate to another substrate. So this cofactor can involve directly in the chemical reaction, okay, by transferring electrons, by transferring groups of atoms, okay. So that is the significance of having enzyme in the reaction, okay. So inilah cara dia kan, empat cara ni macam mana enzyme will lower the activation energy, okay. Okay, so next is free energy. Okay, so organism will live at the expense of free energy. Okay, so what is free energy, or G? Is the available energy to do work when temperature and pressure are uniform in a living cell. So living cell, in order for a cell to be alive, okay, the cell has to do its activity. Mestilah kan, mesti cell tu macam-macam lah reaction akan berlaku dalam sel. Okay, so all these activities within the cell requires this free energy. Okay, in order for the cell to do its activity inside the cell. Okay, so for activation energy, every chemical reaction between molecules involve bond breaking and bond forming. Okay, so therefore activation energy, also known as free energy of activation, is the initial energy needed to start a chemical reaction. Okay, is so from the reactant, you have the reactant. Okay, for example, a reactant, you want to convert it into a product. Okay, into a product, for example. Okay, so so this reactant has to have a start of energy. Okay, untuk dia reach transition state, okay, before it is converted into product, macam tu. Okay, so which is the minimum amount of energy that will get the chemical reaction started. So this is another way of defining activation energy. Minimum amount of energy that will get the chemical reaction started, often supplied in the form of thermal or heat energy from the surrounding. So as I said, without the enzyme, okay, present in the reaction can still occur, but at a very, very slow rate. Okay, so at a very, very slow rate. So that is activation energy. Okay, that's why you need enzyme in the reaction to lower the activation energy, because by having the enzyme, it speeds up the rate of reaction. Okay, tak payahlah substrate tu nak absorb thermal energy ataupun heat energy from the surrounding. Okay, so because by having the enzyme, it will lowers the activation energy and speed up the rate of reaction. Okay, ataupun V increase lah, velocity increase, rate of reaction increase. Okay. Okay, so enzyme lowers the activation energy barrier. So as you can see here, you have the substrate, two substrate in this case. Okay, so the substrate will enter into the active site to form enzyme substrate complex. Okay, so substrate enters active site. Enzyme changes shape such as its active site enfolds the substrate. So this is this explains for the induced fit lah kan. So there are certain enzyme where the active site is not exactly the same as the shape of the substrate, but as the substrate binds to the active site of the enzyme, it causes or induces the active site to slightly change so that it fits. Okay, the active site fits perfectly with the substrate. Okay, so so the active site needs to change also, but slightly, so that to make sure that the attachment between the substrate and the active site, they complement each other, macam tu. Okay. Okay, so next, substrate held in active site by weak interactions such as hydrogen bonds and ionic bonds. Okay, Okay, substrate held in active site by weak interactions such as hydrogen bonds and ionic bonds. So kita dah belajar, kan? Tadi kan kita dah cakap apa dia enlarge pula ni. Okay. Okay, so kita dah belajar yang protein, enzyme ni are globular proteins. Okay, kita dah belajar dulu masa chapter 1 tu, okay. Along the amino acid chain, and every amino acid there are the side chains. Okay, so these side chains, okay, will interact, okay, with the substrate through the formation of hydrogen bonds and also ionic bonds. Okay, so the active site here is composed of amino acid, obviously, kan? So the amino acid will have side chains. These side chains, okay, will interact with the substrate through weak bonds, weak interaction. Okay, weak bonds, weak interactions, such as hydrogen bonds or ionic bonds. Okay, so ini yang point yang ni tadi ni, kan? Ni nombor 4 ni, kan? Apart from having cofactors, the ability of the enzyme to form weak interaction also with the substrate. Okay, by having the side chain of amino acid at the active site tu. Okay, so it forms hydrogen bonds and ionic bonds, okay, with the substrate. So active site can lower the activation energy and speed up the rate of reactions. So dalam keadaan ni kita panggil substrate ni akan reach a state, we call it as transition state. Okay, transition state is the state where the bonds within the substrate starts to break. Okay, new bonds starts to form to produce the product in the active site. So now the product will have a different shape that is different from the shape of the active site. So because the product have a different shape, so now the product will be released from the active site to be used by the cell lah, kan? So now, and the enzyme is now free again to catalyze the next reaction. Okay, so that is how the enzyme works lah, kan? The enzyme works and how the enzyme lowers the activation energy. So you should be able to relate this diagram, okay, this diagram with this slide. Okay, how the enzyme lowers the activation energy. So brings the substrate together in a correct orientation, kan? So nampak? So in a correct orientation, macam tu. Okay. Tahu pula slide dia kat sini, kan? So provide a favorable cellular microenvironment, stretches the substrate bond towards the transition state and form temporary covalent bond with the substrate. Okay, so you should be able to relate this point, okay, with this diagram. Okay. Okay, so next, the effects of an enzyme on activation energy. So here you have a graph. You are really familiar with this graph, I guess. Okay, so you have on the y-axis is the free energy, on the x-axis is the progress of reaction. Okay, so you have here the reactant. Okay, so the reactants, the orange purple reactants and the blue green reactants here must absorb enough energy from the surrounding to reach the unstable transition state. So this is the transition state. So as you can see, initially the bonds between the reactant are really stable. Okay, so as the reactant involved in the chemical reaction and reaches the transition state, the bonds within the substrate will start to break. Will start to break and to form new bonds. Okay, that is transition state. So in order for the reactant to reach this transition state, it must absorb as much energy as possible to reach the transition state before it is turned into product. Okay, so as you can see from the graph, the black graph here, the graph that is in black here is it shows you the rate of reaction without enzyme. So without enzyme, the activation energy will be higher. Okay, and the rate of reaction will take a longer time, nampak, a longer time for the reaction to occur. Okay, because the reactant has to absorb thermal energy from the surrounding, heat energy from the surrounding without the enzyme. So without the enzyme, the reaction can still occur, but it takes a longer time and it requires more energy. Okay, the activation energy will be higher. But in the presence of the enzyme, okay, as we can see, it speed up the rate of reaction and it lowers the activation energy so that the reactant can quickly turn into transition state before it is turned into products. Okay, so enzyme catalyze reaction by lowering the activation energy. A ni supposed to be subscript. Every time when you write the A, it must be in smaller letter lah, kan? Because yang lebih kecil, huruf yang lebih kecil akan EA, like that. Okay, so enzyme cannot change the free energy of reaction. Free energy delta G ni will be maintained. Okay, instead the function of having the enzyme is to hasten, speed up, okay, the rate of reaction that would eventually occur. Okay, macam tu. Alright. Okay, so next, so I hope you understand lah. So if you were to be asked, okay, identify a graph with and without the enzyme, you should be able to identify which one, okay, which one is without the enzyme, which one with the enzyme. So without the enzyme, we'll have a higher activation energy. With enzyme, we'll have a lower activation energy here. Okay. So next is mechanism of enzyme action. Okay, so a mechanism of enzyme action, there are two, which is the first one is lock and key model and the second one is induced fit. So we are going to look at for the lock and key first. Okay, so active site of the enzyme and substrate have complementary shapes. Okay, so it is an analogy lah, kan? So the active site will be the active site of the enzyme will be the lock. Okay, so the substrate will be the key. Okay, so the key, the shape of the key will complement to the shape of the lock. Okay, so that's why it is called as lock and key. Okay, so as the substrate binds to the active site of the enzyme, it forms enzyme substrate complex. So when it forms the enzyme substrate complex, it reaches the transition state where all bonds are broken, new bonds are formed. Okay, products will be produced and the product will have different shape, okay, from the substrate. So therefore, because the product shape is different, so the product will be released from the enzyme. So this type of question are often asked in the exam, often lah, kan. So these are the points that you have to include in your answer. So usually for the answer, for the question that asks you to describe lock and key or for you to explain on induced fit, the mark will be usually 3 marks, and 3 marks, 3 or 4 marks. Okay, so for lock and key, these are the keywords that you have to mention. Active site and substrate have complementary shapes. So they they complement each other. Bukan, dia tak sama dengan awak cakap active site and substrate have the same shape. Salah salah. So active site and substrate, they do not have the same shape, but the shape of these two molecules, they complement each other. Okay. And then point ni pun penting juga for you to include. So this is the first point. The second point, it forms substrate enzyme complex. This is another point that you can include. Okay, all bonds are broken, new bonds are formed. Ini you can mention either of these two. Okay, so product has different shape. Product is released from the enzyme. Okay, so so usually it worth this type of question worth 3 or 4 marks. Okay, same goes to when you are explaining on induced fit model. Okay, the question usually worth 3 or 4 marks. Okay, so when you are explaining induced fit, these are the 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 words that I highlighted here is really crucial for you to mention in your answer. For example, again, the active site of the enzyme for induced fit, the active site of the enzyme is not complementary to the substrate. So initially, for example, here I added a new diagram. Okay, dia pun sama jugalah. So the original diagram should be enough jugalah kan. So the shape of the substrate is not precisely complement to the shape of the active site. So but as the substrate enters into the active site, it causes or it induces a slight change in the shape of the active site, so that when the active site changes slightly, it fits it fits the shape of the substrate. Okay, it fits the shape of the substrate. So saya tambah gambar ni, kalau tengok initially for induced fit, the shape of the substrate is not exactly complement to the shape of the active site, but as the substrate bind to the active site, it causes a slight change, okay? A slight change in the shape of the active site. Okay. So yang ini kalau rasa susah nak write conformational change lah, so you can just write a slight change in the shape. Pun boleh kan? So it induces a slight conformational change or a slight change in the shape of the active site, so that the catalytic group on enzyme is in the correct orientation with the substrate. And then breaking and forming bonds, macam tadilah sama point yang macam dengan lock and key. Okay, products are different, has different shape from the substrate. Product is released from the enzyme. Enzyme returns to its original shape and can be reused. Ini point yang penting untuk awak include, for you to include in your answer. As the product is released, okay, the enzyme returns to its original shape, okay, and can be reused again to catalyze the next reaction. Okay, so for induced fit ni yang penting point ni. Point nombor 1 ni, point nombor 3 ni dengan yang last point ni. Okay, so these are the point that you have to mention when you are answering question relating to induced fit model. Okay. Okay, so factors affecting enzyme activity. So far, do you have any questions before we proceed to the next subtopic? So far saya rasa apa yang kita dah tengok ni, you all dah belajar dah kan. Tapi ni untuk recap. As we go along, so untuk sampai point ni, saya rasa you are familiar with all these facts. Okay. But if you have question, you can ask. Okay, so if there's no question, we will proceed to the next subtopic, which is 4.3, factors affecting enzyme activity. Okay. Okay, so factors that affects enzyme reaction. The first one is enzyme concentration. Okay, so if the amount of enzyme is the limiting factor, it causes the reaction to reach its maximum rate of reaction. Okay, so the concentration of the enzyme will affect the enzyme rate of reaction. Substrate concentration will also affect enzyme reaction. So if you add certain amount of substrate to a reaction without adding more substrate, so this can cause the enzyme reaction to reach its maximum rate of reaction. Same goes to temperature. Every enzyme has its own optimal temperature. Okay, optimal temperature to catalyze the reaction at its maximum rate. Okay, but if you go beyond this optimal temperature, it can cause the enzyme to denature. Okay, so same goes to pH. So every enzyme has its own optimal pH to catalyze the reaction. But if the pH is lower, or the pH is too extreme. If the pH is too extreme, it can cause the enzyme to denature. Okay, but if the pH is not at its optimum rate, okay, the enzyme will not catalyze the reaction at its maximum rate. Okay, at its maximum rate. So ada point yang mana kalau untuk temperature, if you increase the temperature further, it can cause enzyme to denature. Tapi kalau if the temperature is low, okay, the rate of reaction will be very slow or the enzyme can be inactive. Tapi kalau untuk pH, kalau extreme pH, it can cause the the enzyme to denature. Tapi ada pH yang menyebabkan enzyme tu juga dalam keadaan inaktif. Effect pH ni usually is reversible. Tapi kalau temperature, irreversible. Once you have denatured the enzyme, you have broken the hydrogen bonds that hold the globular shape of the enzyme. So it can cause the enzyme to denature and the enzyme loses its specific shape of the active site. So when the enzyme loses its shape, the enzyme cannot catalyze reaction. Okay. Okay, so we are going to look at first the first factor, which is enzyme concentration. Okay, so as you can see from this diagram, you have a graph here. The y-axis is the rate of reaction. Okay, rate of reaction. The x-axis is the enzyme concentration. Okay, enzyme concentration. So if in a reaction you keep on increasing the enzyme concentration, and you make sure that the substrate is in excess, kan? Substrate punya concentration tu sentiasa bertambah, sentiasa ada dalam reaction tu. So you will find that the rate of enzyme reaction is directly proportional to the enzyme concentration. Ini dengan syarat substrate tu dalam keadaan yang excess, banyak, ada lebihan substrate. Okay, so syarat ni kalau untuk enzyme concentration, the rate of reaction will keep on increasing macam ni, kan? The rate of reaction is directly proportional to the enzyme concentration if the substrate is in excess. Okay, so as the enzyme concentration increases, okay, enzyme concentration increases here, the rate of enzyme reaction increases linearly, okay, linearly or proportional, because there are more enzyme molecule available to catalyze the reaction. Ideal if substrate is in excess. Substrate is not the limiting factor in this case. Okay, so graph yang straight awak akan dapat macam ni. Okay, rate of reaction is proportional to the enzyme concentration. But okay, if the substrate is the limiting factor, you will get the second graph here. Okay, you will get the second graph here. So as you increase the enzyme concentration, but the substrate is the limiting factor, okay, you have only certain amount of substrate to catalyze, to involve in the reaction. Okay, so the rate of reaction will reach its maximum rate. Okay, the graph would reach plateau, okay, at certain rate. Okay. Substrate concentration is the limiting factor. Adding enzyme does not change rate of reaction. There is no more substrate for the enzyme to work. Okay, so you will get the second graph. Okay, the more that you add the enzyme in the reaction, but if you have substrate as the limiting factor, the rate of reaction will reach its maximum rate. Okay, rate of reaction will reach its maximum rate, or we call it as Vmax. Kat sini kan, maximum rate of reaction, maximum velocity. Okay, so that is for enzyme concentration. Second, for substrate concentration. So we have this graph, okay, where we have 3 points. So we are going to look at for each point here. Okay, so for the first point, point A, okay, what happened at point A? Rate of reaction increases. Okay, rate of reaction increases with increasing substrate concentration. So a very low amount of substrate here, okay, will cause a very high rate of reaction to occur. Okay, because you have plenty amount of enzyme available. Okay, plenty amount of enzyme, plenty amount of active site available for the substrate to bind. So that's why rate of reaction you see at this point, point A is really high. Okay, so small amount of substrate will bind to the available active site. Okay, active site of enzyme, because at this point enzyme you have plenty amount of enzyme. Okay, because there is more substrate binds to the active site of enzyme. Second, at point B, okay, rate of reaction increases but at lower rate. Kan? So tengok kat sini, rate of reaction will be lower, okay, because less active site is available. Okay, so kat point ini dah sebanyak mungkin substrate bind kepada active site. Bila dah reach point B ni, rate of reaction will start to decrease because less active site is available. Less enzyme are available to bind to the substrate. As you keep on increasing the substrate concentration, you will reach point C. Okay, so point C is when maximum rate occurs because all active site of the enzyme are saturated with the substrate. Okay, all active site of all enzyme molecules are already occupied with substrate. So in this case, enzyme is the limiting factor. So enzyme concentration in this case is fixed. Okay, you have a fixed amount of enzyme to catalyze the reaction, so you will get this graph. Okay, you will get this graph as you increase the substrate concentration. The reaction will reach its maximum rate of reaction as all the active site will bind to the substrate, okay, in the reaction. Okay, so the third factor is temperature. So here you have 4 points. Point A, B, C and D. So kita tengok yang point A dulu. So A, graph yang dotted ni, kan? The dotted graph here. Okay, so it says here, as temperature increases, enzyme stability decreases. So as you increase the temperature, okay, so it can cause the molecule to vibrate. The molecule, the enzyme molecule, or the collision between the enzyme and the substrate to increase. Kan? So that's why when it says here, as temperature increase, enzyme stability decreases. When you increase the temperature, can you keep on increasing the temperature, it causes the molecule of the enzyme to vibrate. So as the temperature increase further, the molecule of the enzyme will vibrate more violently, more vigorously, and can cause hydrogen bonds, the weak bonds, to break. Okay, to break. So that's why cakap kat sini, the stability of the enzyme decreases, okay, due to bond breaks and changing shape of the active site that could lead to the enzyme being denatured. Dia akan masuk point D tu, where the enzyme denatures. Okay, so point B here, kan. So as temperature increases, you have more collision. Okay, there will be more collision between substrate and enzyme. Okay, when you have more collision between substrate and enzyme, you have more formation of enzyme substrate complex and more production of the products. Okay, until dia akan reach point C lah, kan. So point C kita akan tengok sekejap lagi. So for every 10 degrees Celsius rise in temperature, the rate of reaction double. Okay, so yang ni kita akan tengok yang Q10 tu nanti, kan. So for every 10 degrees rise, the rate of reaction will double, will speed up twice. Okay. Okay, so point C is optimum temperature. So balance between enzyme stability and kinetic energy. So rate of reaction is maximum and enzyme is in active form. Okay, is in active form. Okay, so you have the stability between the enzyme stability. Enzyme is in the stable condition at its optimum condition. Kinetic energy is also optimum. So you have rate of reaction is at its maximum. Okay, so enzyme is really active at this state. Okay, so this is the optimum temperature. Okay, optimum temperature, rate of reaction is maximum. Okay, point D, if the temperature go beyond the optimum temperature, the kinetic energy will be higher, okay, and cause the molecule within the enzyme to vibrate vigorously and can disrupt the 3D structure of the enzyme. The enzyme is now denatured, so the enzyme loses its globular shape. Okay, its specific shape. So when the enzyme loses its specific shape, its globular shape, so it changes the active site, the shape of the active site. So substrate can no longer bind to the active site and cause the reaction to stop. Okay, so rate of reaction will stop as the enzyme denature. Okay, so point awak kena tahu lah point A apa jadi kan, as you keep on increasing the temperature, it decreases the enzyme stability. Point B, as you increase the temperature, it increases the collision between the enzyme and the substrate until it reaches the optimum temperature where you have the stability between the enzyme and also kinetic energy. Reaction rate is optimal, and then but if you go beyond, it can cause the enzyme to denature, okay, because the enzyme loses its globular shape, its 3D structure, okay. Okay, so next is temperature coefficient, okay, Q10. So Q10 is the factor by which the rate of reaction increases. Okay, the rate of reaction increases with every 10 degrees rise in temperature. So setiap kenaikan suhu 10 degrees tu, dia akan menyebabkan rate of reaction to increase. By how much? Macam mana nak tahu by how much? You use this formula, okay, which is temperature coefficient Q10. So if you use this formula and you get the value 2, kan, if Q10 is 2, it means that for every 10 degrees, the temperature will, the rate of reaction will double, kan. Speed of the reaction akan naik 2 kali ganda. Macam tu. Setiap 10 degrees kenaikan temperature tu, rate of reaction akan naik 2 kali ganda. Kalau contohnya awak dah guna formula ni, Q10 value dia 2. Tapi kalau Q10 dia Q10 dia 3 lah for example, kan. So maksudnya every 10 degrees rise in temperature, the rate of reaction will triples, macam tu. Okay, instead of kalau Q10 3, kalau Q10 2 doubles, kalau Q10 3 triples, macam tu lah and so on. For every 10 degrees rise, macam tu. Okay. Okay, so here I added a few example, a graph, okay, where you have two different enzymes. So every enzyme has its own optimal temperature. For example, our body, and the human body, where the optimum temperature is 37. Okay, so it means that the optimum temperature for enzyme in our body is around 37. Okay, the optimum temperature for enzymes in our body is around 35 to 38 lah before the enzyme denature. So sebab tu lah tak boleh suhu badan awak yang demam tu tak boleh nak langkau lebih pada ni. So this will interfere with the enzyme activity in your body. Okay, so another example here, you have optimal temperature for enzyme of thermophilic bacteria. So thermophilic bacteria are bacteria that lives in really hot spring, kan, hot spring. Air kolam panas contohnya, kan. So for this bacteria, they can tolerate a really high temperature, where the optimum temperature for this bacteria is around 70 to 77 degrees Celsius. Okay, but if you go beyond, somehow the temperature of this enzyme it can cause the enzyme of this bacteria to also denature. Okay, so for bacteria that, for example, lives in really cold temperatures such as in the Arctic or Antarctic, so the optimum temperature for this bacteria will be lower lah, kan, sebab suhu kat sana sejuk. Kan, bila suhu kat sana sejuk, Arctic and the Antarctic, so for this bacteria, the optimum temperature will be very, very, very low. Okay, but if below than the optimum temperature here, below than the optimum temperature, the enzyme is not denature, but the enzyme will be, kalau kat point yang sini contohnya, kan, if for our human body, the enzyme within our body, if you were to put the enzyme in really cold temperature, such as 0 degrees Celsius, the enzyme is in active state. Okay. But the enzyme is not in nature, but inactive. Kan dia tak boleh nak catalyze reaction because apa? Tak ada kinetic energy tu tadi. There is no kinetic energy to cause collision between enzyme and the substrate. So therefore, there's no reaction. Okay. If the temperature is really low, there is no collision. There is no kinetic energy. There's no collision between substrate and enzyme. So therefore, there's no reaction occurs. Okay. Okay, so next is the next factor is pH. Okay, so pH, every enzyme again will have their own optimum pH, such as for example, the pepsin. Okay, pepsin, which is enzyme in the stomach, will have a very acidic pH to work. Okay, very acidic pH, which is around 2. Then around 2, it requires HCl. Okay, the acidic condition in the enzyme that will activate the enzyme pepsin. Initially, okay, your stomach, the lining of your stomach will produce the inactive form of enzyme. Okay, we call it as pepsinogen. Okay, so this pepsinogen is in inactive form. Okay, inactive form. So in order to activate the pepsinogen, this enzyme, it has to be activated in the condition that is really acidic, which is in the presence of HCl. So in the presence of HCl, pepsinogen will turn into pepsin, which is the active form, which is the enzyme pepsin is used to break down protein. So the digestion of protein occurs inside your stomach using the enzyme protein. Okay, so using the enzyme pepsin. Okay, so as your food enters into the intestine, so the pH of the enzyme that works in the intestine will be different. Okay, it will be more alkaline. Okay, pH of enzyme that works in the intestine will be more alkaline, around 8 to 9. Itulah. Okay. So optimal pH of trypsin, for example, is 8. Okay, optimal pH is 8. Okay, which is enzyme in the intestine. So optimum pH is the surrounding pH where enzyme is most active, and the rate of reaction is maximum. Then maximum rate of reaction. Okay, changes in pH will lead to changes in the concentration of hydrogen ion and also hydroxide ion here. Okay, if you change the pH to acidic, pH 2, you will increase the concentration of hydrogen ion. Okay, if you were to increase the pH, the hydroxide ion concentration will increase. Okay, so changes in pH changes the ionic charges on the active site and substrate molecule. So kita dah belajar tadi, saya dah mention the enzyme. The enzyme is made up of globular protein. Okay, globular protein. Okay, so which is made up of chains of amino acids. So this amino acid will have side chain dia R So the interaction of the R group with one another form the globular structure of the protein, and also form this cellular microenvironment of the active site. Okay, so the presence of the side chain at the active site, this side chain, as we looked before, will interact with the substrate in the reaction. If you were to change the pH of the surrounding, for example, if the pH is too acidic, there is excess of hydrogen ion in the surrounding. Sepatutnya untuk enzyme ni, keadaan dia mesti pH 8 contohnya. Okay, to make sure that the shape of the enzyme is hold together. But if you were to lower the pH, then you will have excess of hydrogen ion. So the presence of this hydrogen ion will disrupt. For example, it will interfere with the interaction of side chain at the active site here. Kan? So it will disrupt, for example, hydrogen bonds that hold the active site. Macam tu kan. So if you have excessive hydrogen ion due to the pH that you lowers, you decrease the pH, you have excess hydrogen ion. The excessive presence of hydrogen ion will can disrupt the ionic bonds. Okay, can disrupt the ionic bonds that holds the structure of the active site. Kan? So it says here break weak bonds. Okay, dia akan melibatkan ionic bond to break. For example, ionic bonds between the side chains of amino acid in the active site to break. So this can lead to the enzyme to be denature, kan? Altering the 3D shape of the active site can lead to the enzyme being denature, kan? So so when you change the shape of the active site, the enzyme denatures. So therefore, obviously, substrate cannot bind to the active site and less product will be produced. Faham So sebab itu peranan pH. Bila awak ubah pH sesuatu enzim tu daripada yang sepatutnya, awak akan ubah dia punya hydrogen ion concentration and hydroxide ion concentration. Bila awak ubah charges ni dalam surrounding aqueous solution of that reaction to occur, and you ubah the amount of positive charge, you ubah the amount of negative charge through the presence of hydrogen ion and hydroxide ion, you will interfere with the side chain interaction of the enzyme, especially at the active site. If you interfere with the side chain interaction, especially the one that involved in the formation of ionic bond, it can cause the ionic bond to break. So this can lead the enzyme to being denature. It changes the shape of the active site. Okay. Okay, so we are going to continue to the next subtopic. Okay. Okay, so the next subtopic, which is enzyme kinetics. Okay. Okay, so what is enzyme kinetics? So enzyme kinetics is the study of reaction rate of biological catalysts. Okay, so this is just a definition. Okay, so in case of if the question were to ask you to define enzyme kinetic, you should know lah. Is the study of rate of enzyme reaction, enzymatic reaction. Okay. All right. So you have this graph here. Okay, so this graph it plots, okay, to show you the rate of reaction. Okay, rate of reaction. Kita wakilkan dengan velocity, kan, velocity v. So y dia adalah velocity lah, rate of reaction. Okay, velocity. X axis adalah substrate concentration. Okay, so it says here, in an enzyme catalyzed reaction, a hyperbolic curve is obtained. Hyperbolic curve is a graph like this lah, kan, where you have a curve graph like this. Okay, so if velocity v is plotted against substrate concentration, okay, S, substrate concentration. So V versus S, substrate concentration. So there is if there is a large excess of substrate, so you keep on increasing the substrate concentration. So the substrate is in excess and the enzyme concentration is held constant. So in this case, okay, enzyme is the limiting factor. Okay, enzyme is keep constant. You have a fixed amount of enzyme. Okay, so this is known as saturation plot. Okay, saturation plot. Why? Because the enzyme is saturated with substrate. You have a fixed amount of enzyme, but you keep on increasing the substrate concentration. So you will reach a maximum point where we call it as maximum velocity. Maximum velocity of Vmax is when all of the active site, okay, all active site of enzyme is occupied with substrate. Okay, is occupied by substrate. That is Vmax. Okay, so you will get a saturation plot due to you have a fixed amount of enzyme, but you keep on increasing the substrate concentration. Okay, so at the beginning of the reaction, okay, at the beginning here, macam kita tengok yang point untuk substrate concentration tadi kan, you have point A, point B, point C tadi kan, okay. So at the beginning of the reaction, at a very low substrate concentration, the velocity of the reaction, okay, rises almost linearly with the increasing substrate concentration. So as you keep on increasing the substrate concentration at a very low amount of substrate, the rate of reaction will increases almost proportionally, almost linearly. Okay. So as the substrate concentration increases, the velocity of the reaction reaches its maximum rate. Yang rate C tadi yang kita tengok tu tadi kan. Yang point C tu tadi. Okay. So as you keep on increasing the substrate concentration but you have limiting enzyme concentration. Okay. Limiting enzyme concentration. So the rate of reaction will reach its maximum rate. We call it as Vmax. Okay, maximum velocity. All active site is occupied by substrate. Okay, so this is because the enzyme concentration becomes the limiting factor. So addition of more substrate will not increase rate of reaction because all active site of the enzyme are saturated with the substrate molecule. So no use lah. No use of you increasing the substrate when you have a fixed amount of enzyme. Okay, so that's why you will get a saturation plot. Okay, saturation plot or a hyperbolic plot. Okay, so as you keep on increasing the substrate concentration, it reaches a maximum velocity. Rate of reaction reaches maximum velocity as all active site is occupied with the substrate. Okay, enzyme is saturated with substrate. Okay, so Michaelis-Menten kinetics. Okay, so Michaelis-Menten kinetics is a model that explains how enzyme enhances reaction. Okay, rate of reaction. And then how rate of reaction depends on concentration of enzyme and substrate. Okay, so in this graph, you will see in the Michaelis-Menten kinetics, you will have initial velocity, VI. Initial velocity, okay, initial velocity which is moles per time, kan? Rate, sebab kita nak tengok rate lah, velocity kan mesti per masa, okay? Rate mesti per masa. Okay, and then you have this is on the y-axis. On the x-axis, you have the substrate concentration. Okay, substrate concentration are measured in molar, kan, molarity. Okay, millimole per liter in this case. In this case of in this graph lah. So Vmax, okay, Vmax is the maximum velocity. Maximum velocity here, okay, occurs when enzyme is saturated with substrate. So as you keep on increasing the substrate concentration, rate of reaction will be constant. Okay, because rate of reaction is at its maximum. All active site is occupied with substrate. Okay, so from there you can determine from Vmax ni, kan, from the maximum velocity, you can determine the value of Km. What is Km? Michaelis-Menten constant. Make sure you write capital K with capital M. M tu tulis dia kecil, subscript lah. Okay, so Km ni is substrate concentration at half Vmax. Okay, so basically Km is Michaelis-Menten constant, where it indicates, okay, how much substrate is used at half of the maximum velocity, kan? So maximum velocity roughly kita tengok kat sini around 100, kan? Around 100, okay, roughly. So it means that the value for for Km, the value of Km, kan, substrate concentration, is when the Vmax is half. Half of Vmax is 50, kan? So half of Vmax is 50. So from there you can know the value of km, which is around at this point here, around 10 kan, 10 millimole per liter macam tu. Okay, so it means that for this reaction, the reaction only requires a very small amount of substrate to make sure that the reaction that the enzyme can react with the substrate macam tu kan. So the Km value will determine whether the substrate is suitable for the enzyme or not. Okay. Okay, so Michaelis-Menten constant Km is the substrate concentration at half Vmax kan. So tadi kalau maximum velocity is 100, so half Vmax will be 50. So tarik graph dia ke bawah untuk determine the substrate concentration, that is the Km value. Okay, Km value at half Vmax. Okay, so it's a measure of the affinity kan. So basically, why do you need to determine the Km value? You want to know, okay, whether, okay, the enzyme can react with the substrate or not. Okay, so it's a measure of the affinity of enzyme for its substrate. Okay, so if this the smaller the Km value, it means that the higher the affinity of enzyme for its substrate. Macam kita tengok tadi kan, graf ni tadi, Km value dia 10. Maksudnya you only requires a very small amount of substrate so that the substrate can quickly react with the enzyme, can quickly react with the enzyme. So it means that the substrate will have higher affinity for the enzyme. So Km, the smaller the Km value, the higher the affinity of the enzyme, of the substrate to bind to the enzyme. So you want the substrate with the lowest Km value. Okay, you want the substrate with the lowest Km value because there you will know that the rate of reaction is really high. Okay, so it says here the smaller the Km value, the higher the affinity of enzyme for its substrate. So lower substrate concentration needed to reach half of the maximum velocity of that reaction. Okay, so the more strongly the enzyme binds to the substrate, the more likely the product to be formed. This is dengan keadaan Km value tu kecil. Kan? When the Km value Michaelis-Menten constant Michaelis-Menten constant is low, the enzyme will react quickly with the substrate. Okay, because the enzyme will bind more strongly to the substrate and more product will be produced. When you have more product will be produced, so it means that the enzyme will have higher turnover rate, kan? Higher turnover number, Kcat ni. Okay, which is the maximum number of substrate molecule converted into product per second. Okay. Okay, so further explanation on Km. So Km, again, you have to know it is a measure of substrate concentration requires for significant catalysis to occur. Okay, so dengan awak tahunya Km value ni, you will know that the rate of reaction is really high because the enzyme has higher affinity for that substrate. Km values for a particular enzyme depends on the identity of a substrate, temperature, pH, and presence of inhibitor. Okay, so Km values are influenced by this factor: identity of the substrate, temperature, pH, and presence of inhibitor. For example, okay, if an enzyme can catalyze a reaction with two similar substrates such as, for example, glucose and fructose. Kita dah belajar chapter karbohidrat hari tu. Glucose and fructose, they are isomer of each other. Okay, they are structural isomers. Structural isomers kan? Sorry, dia molecular formula dia sama tapi structural formula dia berbeza kan? Sebab kalau glucose ni dia punya carbonic group dia at carbon number 1, fructose carbonic group dia at carbon number 2 kan. So they are isomer but the structure is different. Okay, so it will prefer a substrate. So this enzyme will catalyze the reaction with the substrate that has lower Km value. So dia nak pilih yang mana? Dia nak pilih glucose ke dia nak pilih fructose? So enzyme tu akan tengok Km value of fructose and Km value of glucose. So if the Km value of glucose is smaller, the enzyme akan prefer bind kepada glucose instead of fructose. Okay? So can compare efficiency of different enzyme for same enzyme or source. Kejap ya saya. Okay. So therefore maximum velocity, the rate of reaction when the enzyme is saturated with substrate. So that is Vmax. The rate of reaction when the enzyme is saturated with the substrate. Yang tadilah kan maximum velocity tu tadi. Kan Vmax. All the enzyme active site is saturated with substrate. So reveals the maximum number of substrate molecule converted into product per second. So I leave it you to this slide dulu. Tolong fahamkan kenapa kita nak tahu Km value ni. Slide ni sangat penting untuk fahamkan. Before we proceed to the next slide, saya nak kena settlekan anak saya kejap. Fahamkan ni dulu, Km value ni. Okay. Okay, so we continue. Okay, so that's why you need to know the Km value, where the Km value will determine whether an enzyme can catalyze a reaction with that substrate or not. Okay, so the enzyme will choose the substrate with the lower Km value because the substrate with the lower Km value will have higher affinity to bind to the enzyme. Okay, so therefore the enzyme can catalyze the reaction at higher rate. Okay, so can compare efficiency of different enzyme or same enzyme of different sources. Okay. Okay, so Vmax, the rate of reaction when the enzyme is saturated with substrate. Okay, reveals the maximum number of molecules converted into product per second. Okay. Okay, so this is the graph. Okay, it says here the value of Km are measured in terms of molarity because Km is actually the substrate concentration. Okay, Km is the substrate concentration at half of the maximum velocity. Okay, at half of the maximum velocity, that is Km. So that's why Km is measured in molarity. It is the substrate concentration at half Vmax. Okay, so typically the values of Km for most enzyme range between 1 millimole to 1 micromole. So the range of the value for you to determine the Km is really small. Okay, so for you to determine Km value using this graph is a bit difficult. Okay, so when you are using this graph and the hyperbolic graph here to determine the Km value that you obtain from this type of graph is just an estimation. Okay, because the range of value is really small. Okay, so for you to determine the Km value using this hyperbolic graph is just an estimation. Okay, so you will need a really large amount of data. Okay, to plot this kind of graph. Okay, to plot this kind of graph you need a really large amount of data. Okay, so you need to carry out many experiments to get the data. So it is also more difficult to determine Vmax value because the gradual upward slope of the hyperbola. So the gradual, point mana sebenarnya orang nak stop kan, sebab dia punya rate dia lebih kurang bila dah start reach Vmax tu. Okay, so when you are using this hyperbolic graph to get the value of Vmax and Km, it is just an estimation. Okay, so therefore you will convert this hyperbolic graph, into a more linear form. Okay, into a more linear form, a straight line form, which is, we call it as a Lineweaver-Burke plot. Okay, so Lineweaver-Burke plot is when you take all the values from the previous graph and you reciprocal the values. Okay, it is a double reciprocal plot where you 1 over kan semua value untuk velocity and also substrate concentration kan. So at y-axis it is 1 over v. At at y-axis it is 1 over v. At x-axis it is 1 over substrate concentration. So semua value yang initially dalam graph ni awak satu perkan dia. Okay, to get the straight line. Okay, to get the graph in in a straight line form, okay, which is Lineweaver-Burke plot. Okay, so this so it is a straight line plot will be obtained. So this provides a more precise, okay, a more precise way to determine the value of Vmax and also Km. So how do you determine the Vmax and Km value? You look at the intercept at y-axis and also at x-axis. So the intercept at y-axis here, okay, which is 1 over v, you will get the Vmax value, kan? Value yang awak intercept kat sini, you will get the Vmax value, okay? At x-axis here, kan, dia akan pergi dekat side yang lebih negatif, kan? So you will get the Km value. So intercept, awak kena letak sentiasa formula dia as negatif Km sebab dia mesti pergi ke belah yang lebih negatif. Bila awak plotkan dia, awak kena extendkan garisan tu supaya you get that intercept. Okay, so it says here, I added a bit more information. It says here the Lineweaver-Burke plot will transform the hyperbolic plot, which is Michaelis-Menten kinetics, into a linear plot. So it is an easier and more accurate method by using fewer data points to plot the reciprocals of the velocity and substrate concentration against one another. Okay, so you use this graph, Lineweaver-Burke plot, to get a more precise value of Vmax and Km. Okay, so you have an exercise here. Okay, so in this graph, okay, in this table, it shows you it gives you a value for substrate concentration. Okay, and it gives you the value of the rate of reaction, the velocity. Selalunya dalam soalan dia akan bagi unit tau. Okay, so make sure when you are answering the question in your exercise, or for example if it come out in the exam, for example, kan, you must write the unit. Do not leave out the unit in your answer. Okay, in your graph. In this case, sebab dia ni adalah latihan, kan, so tak apa. Dia latihan, kita boleh ignorekan dia. Tapi make sure when you are answering this question, you include the units. Okay. Kita ada co-lecturer kat sini. Kejap ya, you have to bear with me. Okay. So latihan yang pertama daripada table ni, you have to draw a graph. Okay, draw graph velocity versus substrate concentration. So from this value, you should get a hyperbolic graph. You should get. So make sure after this lecture, you do this exercise. Okay. So for question number 1, make sure you get a hyperbolic graph. So for question number 2, okay. For exercise 2, draw a Lineweaver-Burke plot. So in order to okay, in order for you to get the Lineweaver-Burke plot, okay, macam mana cara you tell me macam mana cara untuk dapatkan graf ni? Sebab sebelum anak saya tanya, baik aku beritahu.
Buat 1/2.
Awak over kan setiap value ni. Faham? Setiap value ni, make sure you 1 over kan, get the full value. Lepas awak dah 1 over kan setiap value. Cuba buat satu value, 1 over 0.02 dapat berapa? You should get a whole number.
50.
50 kan. So you do the same for all these, and then you plot the graph. So when you are plotting the graph, you should get a straight line. Okay, you should get a straight line. So kat mana dia intercept ni kan? Dekat y-axis ni. Kan, bila dia intercept dekat y-axis, you can determine the Vmax value, kan? Kan, mana dia intercept dekat sini, 1 per Vmax. From this value, you can determine the value of maximum velocity. Kat mana dia intercept dekat x-axis, you can determine the Km value. So make sure bila km value kan formula dia 1 per km, value sebelah sini pun negatif kan, so nanti dia akan cross out each other lah kan, dia akan cross out each other, so you get the value. So make sure don't leave it as whole number macam tu, make sure you write the unit. Sama juga after you got the value for maximum velocity, you write the unit. Apa unit dia? Mesti per time kan. So whatever time lah dia bagi kat situ. Okay. So ini saya bagi ini awak boleh, ini saya bagilah kan. Ini saya bagi. Sepatutnya kalau awak plot, you should get a hyperbolic graph like this. Okay. And for the Lineweaver-Burke plot, after you convert all the values dekat graph tadi, 1 over kan tadi, you should get a whole number and you plot the graph kan. So you should get a straight line. Okay, so daripada situ determine the value of V max, okay, and the value of Km. Faham eh? Do you understand?
Yes.
Okay, so ini you can do in your own time. I'm not sure if ada latihan ni tak dalam soalan workbook. Saya tak go through in detail lagi. Sekejap saya tengok. Tak ada pula. Tapi tak apalah. Tapi make sure ini sebagai latihan awak lah untuk awak fahamkan kan. Okay, awak boleh cari other exercise that has this sort of activities. Okay. Okay. So that's I guess we are going to stop until cofactors.